también el postdoc, y hoy en día es donde se desarrolla y se desenvuelve como full professor, which is not mine, it's at the top, right? Full professor. Daniela tiene una serie de trabajos bastante relevantes en lo que es levadura, desde una perspectiva bastante de biología molecular, los RNAs, pasando también por aspectos más ecológicos, y hoy en día algo que desarrolla bastante, un poco por el financiamiento que está teniendo actualmente Inglaterra, son una lista más biotecnológica, donde you've got projects with ABBEV, colaboraciones con algunas cervecerías, un poco también para apoyar el financiamiento del laboratorio, teniendo una industria biotecnológica bastante importante. Entonces, hoy día, bueno, nos va a contar un poquitito acerca de esa historia, y muchas gracias a ustedes por estar acá. And thank you very much, Daniela, for taking the time to be in the seminar and to come to Chile.
Thank you very much, Francisco. I didn't understand the word. Don't believe what he says. I'm actually a good person. But anyway, today I've been asked to show you a little bit of the research that we are doing on the more sort of biodiversity and biotechnological side. So I just want to, focus on that. How can we harness this biodiversity and use it for biotechnological purposes? So,
biodiversity, what do we have? This is the Saccharomyces genus and the different rearrangement which happened over the year. And the first time the Saccharomyces name was coined was in 1898, Mayan. It meant sugar mold. And then there has been a lot of species put in this sort of under this genus, mainly distinguished by morphology. But as the molecular tools became more available, restriction fragment length polymorphisms at the beginning, and then the sequencing, we managed to restrict the Saccharomyces genus, and in 2003 there were about 7 species. What happened over the year is that new species were found. Saccharomyces micratum cuirezevi were defined and find as new species in 2003. In 2011, we had another species being found, which is arboricola. And then the last species genus which has been added is, Eubayanus here, most important, and the last species to be added is Saccharomyces urei. And Saccharomyces urei has been discovered in my lab, and I'm going to tell you a little bit what happened there. So basically, it was for us, it was like a hobby project. At that time, we were very much focused on gene function and understanding the role and the function of the gene in different environments. But I was also, unfortunately, leading a group of second year undergrad students to do their field course in France. So now this misbehaved. Okay, so that's where we were. So we were, here is Nice, and we had a little short journey up to the pre-Alps here in this lovely Saint-Aubin. This is 1,000 meter altitude, and actually there are some oak trees at that altitude, and it's quite rare to find oak tree at such a big altitude. And the students, as you can see, the last day, they do what they want. They study 2 weeks really hard. The last day, they go canyon. And the staff, me, my postdocs, decided to go and collect some bark and soil. Let's see what happens. So that's Samina here, which is sort of collecting and showing with the finger the first tree, second tree, and so far and so on. So then we, let's see if this comes up. Yeah, so we got some classic bark, we got some colonies, but I got to say, this has been done really naively. We came back to the lab, we didn't know what to do. We asked a colleague, put in some media, put in Synagogsky media, put it in there, let it grow. So we got some colonies, I think Samina forgot them there. And we had about 200 potential yeast colony. So the next thing is, we said, what do we do? Let's send them to Norwich. In Norwich, there is this National Yeast Culture Collection, and they were expert in in defining what it is. So we sent 3. We sent 3 samples of oak, a couple of soil in a bark. They did an ITS and they said, well, one is Kazakhstania cerevisiae, but this one here is weird. I mean, it looks like Mikari, but it's not Mikari. It's 98% sequence identity, but in the ITS, but it's just different. It's not quite Mikari, and it's not quite Paradoxus. And I said, oh, maybe something new. Bah, maybe. And they said, anyway, there is nothing you can do about it because you need at least 2 specimens to actually be defining a species. Fine? Samina, send another 5 colony. So that's what she did. So she sent another 5. Let's see if this come up. That's it. So she sent another 5 colony, and sure enough, this time on a bark, something, I said, these had the same ITS as this one, but this time, you know, as you put in the software, it came out more closer to Paradoxus. And again, I said, is this novel maybe? And I said, bah, I don't know. So they did an ITS 3 with the 2. No, why this is not so prompt, okay. So, and this species kind of came here, close to Paradoxus and cerevisiae, a little bit close to Mikari. So I said, okay, let's send all 200. All 200, off you go. So we sent 200, and all the rest were either Paradoxus or they were Kazakhstania cerevisiae. There was no more. So we were very lucky in the first 8 samples out of 200 to actually find the 2 new species. So at that point, we decided to sequence the whole genome. And first of all, we look at the COX2 and COX3, and it was very close to Paradoxus. Only 2 differences here for COX2, 3 different COX3. Did a 3, and it came sort of close to Paradoxus. In reality, when we did the entire sequence, well, first of all, we realized that there were a few translocations. There were 2 translocations reciprocal between chromosome 1 and 13 and 6 and 7. So if you look at the dot plot, you can see there is 1 translocation here and 1 translocation there. Interestingly, the translocation in the middle is the same translocation that you have in Mikati 1815 and 1816. So this Saccharomyces uri, as we call it, has 1 translocation in common with the Mikari, and it's got an extra one in the genome. So then we looked at the phylogeny, and as you can see from the phylogeny, we concatenated 100 conserved gene, and now you can clearly see that the 2 isolates comes close to Mikari. And basically, you wouldn't have made that assumption if you look at the mitochondrial gene, and you wouldn't have made that assumption if you were looking at ITS, because that would look closer to actually Paradoxus. So, Jesus, okay. So, are they different biological species? So, what we did, we crossed the uri, the haploid uri with cerevisiae. We didn't do this blindly. We actually created all the tools. We created an HO deletant, and so the strain were heterotelox, and they could mate. Then we get some diploid hybrid. We select them on the kanamycin, and then we basically look at the sporulation. Do they sporulate? How many spore we have? So the first cross was with cerevisiae, and the spore variability was extremely low for both strains. Second cross was with Mikari. 2% was the best we could get in this case. And then we tried all 3 populations of Paradoxus, the South American, the Middle East, and the European. And as you can see, both for one species and the other, for all 3 Paradoxus populations, the maximum you can get is 1.6. So at this point, there was a very strong indication that Saccharomyces uri is indeed a new species. And that is how it was inserted in the in the phylogenetic tree. Since that time, I know other groups and found URI in Bavaria, the group of Brian Gibson, and Jenny Litton itself. He went and found few more URI around Europe. So, of course, we were really quite galvanized about these results, and well, let's go and do bioprospecting of the Alps or whatever. So again, because I was going with the students, second year undergrad, to do a field course, we were to Italy. So this time we went to the Alps here. And the Alps, again, we had different altitude, and we were sampling bark and soil. We were putting different selected media, we tried different temperature, we tried different enriched media, so we were all quite excited, maybe we'll find something. Everything that we found here was Paradoxus. So I got a little bit frustrated. I said, well, it may be that the way we're actually isolating our yeasts is really skewing towards Paradoxus. At that time, also, Duncan Gregg did a very nice work looking at a metagenomic signature on barks and oaks to see the community of the Saccharomyces. And he came up with a surprising finding that actually there isn't many yeast at all in bark, because the majority is Basidiomycetes. Reading that paper, I just thought, yeah, but what Duncan did, he amplified the ITS and he sequenced everything. So if you have a lot more Basidiomycetes, you're going to dampen the signal. You're not going to see very much. So what we did, we decided we can amplify the ITS region, we can run it on a gel, and we know that for the Saccharomycetes, the ITS band which we are expecting to see is 820, about 100. Other fungal species will have a much smaller ITS. So the idea here was done for the metagenomics studies, take different samples from different trees at different altitude, extract the DNA from the soil, amplify the ITS, but then you run the ITS on a gel and you specifically cut out from the gel a PCR product which is above 800. So in that case, you are sure you're enriching for the community you want to see on the tree. And that's what we did, and to make a story short, we had some oak, beech, and spruce at different altitudes, some pine, and clearly, well, the first things to say is that we managed to see all these species as a metagenomic signature, except for Arboricola. So the Saccharomyces species reads were much more noticeable, the abundance was much more on oak and on soil of tree at the same sampling location. So, so basically, there was a strong sort of idea that oak probably is a natural sort of habitat for the Saccharomyces. And the other thing, Paradoxus actually, although we isolated consistently only Paradoxus when we are screening the sample, it was not at all the most abundant species in our metagenomic reads, because the most abundant was Kudryavtsevi. It was very abundant everywhere. And Paradoxus was less abundant even of Kudryavtsevi and also Mikari. So really, there must be some skew when we do the isolation methodology. And finally, I think this is very similar to what I've been seeing in Francisco's group's data. When you look at the altitude, and that you can correlate pretty well with the cold or harsh environment. If you look at the species abundance at 600 meters versus 1,400 and 1,800, you clearly have a decline of species that you can see that. So the little conclusion here, I think the high throughput sequence of this ITS1 region, which is amplified only from the 800 specific ITS size, is quite effective to capture the diversity of the Saccharomyces species. And we were able to detect most of the species, including Eubayanus and Micati. So Eubayanus, we had, if I go back, oh, sorry. This is going to be very long talk, because if these I have to do this for, yeah, okay. So Eubayanus, which is this little thing, we found a sample, an OTU, we found in oak, and one in spruce. The other thing which was quite interesting to note is Euvarum, which also we found little, but the only little we found was primarily on beech, which is your Northofagus sort of kind of tree. So it's kind of interesting to notice that. So yeah, admittedly we only have a couple of OTUs for Eubayanus, but they were pretty specific, 100% similarity. They were on both different trees, so running all sort of statistics that came fairly significant. But yeah, hands up, I mean, I'm happy to be challenged on that. So going back to this taxonomy, so we got these all new species, but there was also a lot of species which got kicked out. Scariocanus, for example, introduced in 2003, disappeared in 2011. That is because from the genetic point of view, it's completely similar to Paradoxus. And the reason it was classified as a different species was based on biological species concept. And the reason why he was reproductively isolated is because of the presence of 4 big translocations. So every translocation reduces your spore viability of about 50%. So 50% for 1, 25% for 2, 12.5% for 3. If you got 4, you go to 6.5%. So clearly that was due to the structural variation of these species rather than the genomic variance overall. So this was kicked out. And then there was a big saga about the Bayanus and the Bayanus 2003, then we got 2 variants, Euvarum and Bayanus, and then actually the Euvarum is the only real species because the Bayanus is an hybrid between Eubayanus, Euvarum and Cerevisia. So this is where we stand at the moment. We have 8 true species and we have a couple of interesting hybrids. And so what about the hybrids? Well, the hybrids are really interesting. In fact, we got several hybrids, and the hybridization in Saccharomyces cerevisiae occur usually quite readily. You got hybrids of all sort of type and shape. They've been isolated primarily from domesticated environments, beer, cider, wine. Some are from native trees like olive trees and so far and so on. But the fact, you can already tell the fact that there are so many hybrids connected to human activity, there is a lot of companies and industry which are interested in exploiting this hybrid. So I've been working in the past with AB InBev, with Better Tech Hop Product, Cloudwater, BP, Singer, this is just a high throughput company, and A&B, which is develops strains and distributes strains for wine. So all these companies are interested in developing hybrids for biotechnological purposes. So why would you hybridize? Well, so you can create offspring with high genetic variation. So that's give you a big phenotypic space. Secondly, you can combine advantageous traits from both parents, so the heterosis or the hybrid fitness that you can see in some cases. And also you can create another source of variation for the evolution to act upon. If you think about your finches and El Niños, the hybrids were not really very viable, and then when the El Niño came, changed the seed size in the Galapagos, and then everybody could actually be selected favorably, and all in a sudden the hybrid became established. So these are really important things to bear in mind. And if we look now molecularly what happens when we make a yeast hybrid, so first of all we got two genomes which coexist. It's very simple, and you know well that you got different allele variants and copy number which can affect the phenotype. One allele can be more expressed than the other, or there could be cis-trans differences, expressional changes, and that is already giving you a variation. The second thing which can happen in the hybrid is that actually two proteomes coexist. Yeah, okay, fair enough, you got two genomes, you got two proteomes, but the function in the cell, more often than not, is carried out by protein complexes, not just by one protein. So you can imagine a situation in which members of the protein complex assemble, and this has to be done correctly, to carry out a biological function. So now you can imagine in a situation where you have two homologous proteins for everything, you can have all sorts of assembled protein complexes that can be unispecific or chimeric, and all different scenarios can actually give you a different type of fitness. So again, evolution can play a role in selecting one or these other protein complexes according to the function that they carry. And thirdly, in the yeast hybrids, you have one or the other mitochondrial DNA. So originally when you make the hybrid, you got two nuclear genomes and the two mitochondria, but the two mitochondria is going to be, one of the two is going to be kicked out. Within 20 generations, you either kick one or you kick the other, and you either being left with the gray one or the pink one. And we have now an abundant amount of information that according to which mitochondria you keep, you have different phenotypic and transcriptional landscape. And this is also quite important. Now, how can we use all these natural phenotypic variation to improve, for example, specific biotechnological traits? How can we use these new species that we found? How can we use this new population that we found? And how can we use the hybrids in this term? And this just shows how something can evolve to keep one or the other combination in general. So one of the biggest issue in using interspecific hybrid is the fact that the hybrid in itself is sterile. So an example, if this collaborates, non-collaborates. So this is a classical example from the Aristotelian time. You have a horse, you have a donkey, you have a nice little mule, which is really useful for human activity, but the mule is sterile. You don't have any offsprings. And I think the very same thing happened with yeast. You got Cerevisia, you got Kudryavtsevi, you got very healthy hybrid, you can use these. But these two chromosomes do not recognize each other when they want to go through meiosis. So you don't get any viable offspring. So all you can do is work with a mule, but you can never work with the children of the mule. This is the bottom line. However, as you may know from the plant world, if all of a sudden, instead of having a hybrid which is a diploid, but you have a hybrid which becomes a tetraploid, so for example, you have two copies of Cerevisia and two copies of Kudryavtsevi, all in a sudden you restore the fertility because you restore the fertility because the two cerevisia chromosomes will find each other, even if they are different strain. They could be diverse strain, that's the different gray shades, but they would still recognize each other and separate. And still, and same thing would be for the Kudryavtsevi. So you end up with a progeny with scrambled trait, which is viable. So that's what we did. We tried to create this allo tetraploid. This is going to be really challenging because there is a lot of animation and this isn't collaborating. Let's try. So we took cerevisia strains from North America and, for example, Japan, Kudryavtsevi for Portugal and Japan, which are all also quite different, also metabolically different. One can use galactose, the other cannot. And then we decided to make hybrids. So these hybrids here is this green with this red, and this other is this light green of this red, or whatever combination. So you got inter-specific hybrid. Next things you do, you want to mate these. This is now gone crazy. So to mate these, you need to delete one mating type locus. So if you delete the mating type locus A here, all in a sudden the diploid would perceive itself as a mater because it doesn't have one mating type. So this will perceive itself as an alpha, and if we delete the alpha here, this will be perceived himself as an A. And so you can mate them, and you can have a fertile for hybrid this time, in which you got this chromosome of cerevisia and Kudryavtsevi that can recombine, and therefore you can have a progeny. At this point, you can let the spore germinate, and you will find your F2 generation. Now, if you do a lot of cycle of meiosis with the generation, you can create a very, after 12 cycle, for example, a very much scrambled progeny. So all these traits are really broken up by recombination, and you have a patchwork of the different species and the different strains genome. And this work has been done in my lab in collaboration with Ed Lewis lab, and Ed is, has been a pioneer in quantitative trait locus analysis, and he has made a hybrid with Eubayanus, and and I forgot to say, we also had either So we had hybrid with chromosome from cerevisia and hybrid with chromosome Kudryavtsevi. Sorry, it's just not working. So, and Ed is done hybrid with the cerevisia uvarum and with cerevisia eubayanus. I concentrated with hybrid with cerevisia and Kudryavtsevi and cerevisia and uri. So then what we did, we took all this population and we look at the phenotypic space. And we look at different temperature in different concentration of maltose and at different concentration of acetic acid. And as you can appreciate, the progeny has a huge difference. Some grow really well, some grow really poor. This is a hybrid progeny with the mitochondria from cerevisia, and this is a hybrid progeny with the mitochondria of uri. And so that's the top performer, that's the bottom performance. So by dividing top with bottom, what we were able to do is do some quantitative trait study to see which allele are responsible for the trait that we are studying. So we had, we took the 20 with a high fitness and the 20 with the worst. We had two pool, and we sequenced these two pool with Illumina, and we looked at condition temperature, maltose, and acetic acid in my lab. And what we end up is having this multiplot. For example, in this case, you have the red lines, which shows the allele frequency for a high fitness. So as you can see in this locus, all the high fitness progeny prefer this allele over the green one, and the majority of the low fitness progeny has the green one. So you kind of know that allele is important for that particular trait. And also what we had different hybrids. We had Scudo, Cervice, different mitochondria, and we pulled out the same region in several of them, which was important for that trait. And just by overlapping the region, we were able to see, for example, for the temperature, the COX-6 is coming as in common to all the different hybrids. So clearly must be important for the temperature trait. That is a mitochondrial monooxygenase and is also involved in beta fatty acid oxidation, and again makes sense because temperature, membrane fluidity is related to the amount of lipids you have, and the ratio between short chain and long chain fatty acids, and therefore this kind of fit the idea that that could be important for the temperature. And then we had, for example, for maltose, we found one which was MAL33, which was pretty obvious, and that was in common with a lot of different strains. For acetic acid, we had less QTL overall, but we had a couple of regions which were quite interesting, and we had a bunch of genes that which we tried to look for what could be the causal gene, and QM1 was one of the candidates. So the other thing we look is that what happened with the mitochondria? Do they have an effect? Well, actually, if you look at the QTL in Cervice Kudu hybrid, either with the mitochondria of Cervice or with the mitochondria of Kudu, they are different. So all the red, Cervice mitochondria, all the blue are with Kudu mitochondria, and there is only a little bit of QTL which are in common between these two tetraploids. And again, that shows how much the mitochondria might have a profound effect on the QTL landscape. And a similar situation, very little overlap between the QTL of the two mitotype was seen in Cervice and urea hybrid. So next things, as you probably, if you ever done a QTL study, you need to validate a couple of genes that you found just to see whether is this actually real, does it hold up. So the QTL that we saw were specific to the Cervice alleles in the tetraploid hybrid. So what we did a reciprocal homozygosis analysis. We choose one candidate gene for each condition. And the idea here is to create a tetraploid where you only have one allele and you delete the other, and the other tetraploid where you have the opposite scenario, you have one there and the opposite one deleted. So by comparing these two in growth assay, if they grow the same, well then there is no validation because really there is no difference in these two alleles. But if you see a difference in the growth, yes, you can validate your phenotype. And that's what we saw because although small, as you can appreciate, this was significant and statistically quite significant for all three genes. So the other thing that you need to, we thought, okay, but are allele 1 versus allele 2 manifest, the fitness advantage, is it manifested only on the hybrid? Or even when you have just the Cervicia, you can see this difference. So is it hybrid specific or is it simply species specific? And to do that, all what you do is you want to see how these two behave, but just in a normal Cervicia background. So you a Cervicia hybrid with that particular allele deleted or the other. And again, we ran the same experiment, and this time you can see we had no difference here, no difference there. In here we do have a difference, but it's opposite of what we see here. In this case, the black allele, let's say, is fitter, and in this case, it's less. So what we could conclude here, that the interspecific hybrids generate QTL which are not present in the parental species, and therefore they are hybrid specific. And that again points to the fact that it gives you more variability at for the evolution to act upon. So if you want to evolve, or if you want to select or use this hybrid for biotechnological purposes, you've got a much bigger space to play with. So that's a couple of conclusions. We have extreme phenotypic distribution in segregable interspecific hybrid. The QTL we found dependent and independent mitochondrial origin. We identify QTL which are hybrid specific. So how can we now exploit these in a biotechnological term? And you can imagine if you have this large phenotypic space, what you can do is you screen for the optimal chassis for whatever trait you want. So we are looking now at production of green chemicals and organic acid. We are looking for host which has an optimal grow at low pH. So we are screening all these hybrids, and we're just picking the one which consistently has the best grow at low pH. And BP was very interested in this type of project, and is funded some studied in my lab, and so has Singer. You can also look at biopharmaceutical. So what we did, we screened for resistance or susceptibility this hybrid for different antifungal. And again, here there are fluconazole, miconazole, mecafungin, caffeine, flucytosine. Look at the big space. Some really cannot survive. Some shows quite a bit of resistance. So again, this is we're now finishing this work, and we are validating few targets, which actually finished. William has finished, and we're about to write this up. But basically, it could also give you an insight on resistance and susceptibility to different antifungal. And finally, I just want to show you an example of how we exploit Saccharomyces uree and the hybrids to create a new craft beer in the craft beer fermentation. And as you probably may know, the craft beer market exploded. You know, the whole world became crazy, and they all want to drink alternative type of different flavor beverage. So we had a collaboration with the Cloudwater Brew with Paul and Mark. Paul is the CEO, Mark was the chief brewer, and we got some money from Innovate UK and to carry out this project. So the first observation was that Saccharomyces uree grew very well on different carbon sourcing, and especially grew well on maltose and maltotriose, and these are, you know, the main sugar component of the wort, so that was really good. The second observation is the uree, temperature-wise, this is just the optimal temperature, is close to Eubayanus and Euvarum, and basically it grows pretty well at cold temperature. So again, that was an encouraging trait to try to develop some beers. So we tried just uree, and it was just really not good. It wasn't fermenting well at all. So what we did, we tried to cross it with some other commercial Saccharomyces strain. So, well, yeah, first of all, I got to say my colleague put it in the garage in a couple of things. He tried to brew different bottles, and actually you could tell, even just from this crude garage sort of brew, that the uree had a distinct different flavor. It was a little bit more smoky, a bit more clove, and when we, in fact, we got them made them try this, and so they were really quite, oh, this is different, we can try to do something. So Costantina, who worked in my lab, was the research associate who carried out the majority of this job. That's Costantina. So the first issue here is that we need to do everything non-GMO. So the uree and the Cerevisia ale strain were simply sporulated, and then blindly, we didn't know what we were doing, we put a spore against the other, and hoping that some of these spore-to-spore mating would be correct, an A and an alpha, and will give us an hybrid. So this hybrid, then so we need to do a lot of screening for, with species-specific primer for chromosome and different genes, and also we do fax analysis to look at the DNA content to make sure that they were what we think they were. So Costantina isolated, I think 12 of these, but then we used three others which looked a little bit more promising. And we did some pilot fermentation, and as you can see, this is the gravity, the sugar consumption really, and this is urine. Urine itself is really not good. It doesn't consume sugar, it stays up. The cerevisiae strain is here. This was not ideal either because it was consuming too much sugar and it was hyperattenuating the beer, and the beer became too astringent. But the three hybrids were somewhere along the line, and just talking to the brewer said, you know what, I think the H2 and the H3 are perfect because they have about 80-85% attenuation. That's what we like. We'll try them. So doing this hybrid, the hybrid H2 and H3, they had efficient fermentation and they had optimal sugar attenuation. So they liked that. So the third thing to look is that what type of aroma and flavor this hybrid might produce. So what we did the GCMS analysis and look at the combination of profile. And again, the hybrids had slightly different profile than the parent. There was a little bit more of vinyl guaiacol, which you don't really want too much, but you don't want nothing either. So just talking to the brewer, they want everything in the right amount. I mean, it's not like a yes and no. It's all, a bit of this, bit of that, but not too much of this and too much of that. But we had quite a good different spectrum of aroma profile, which was distinct. Of course, what you see in a GCMS is not necessarily what you smell and what you taste, because there might be some of these compounds might be below your sensory threshold. So we ran some sensory evaluation. This has been done by Cloudwater, by some expert. And this is the normal cerevisia, and as you can see, it was very astringent. They liked the fact that it was estery and that was nice fruity, but it also was very astringent. Uri was less astringent, but it was a little bit too phenolic. So again, in itself, it wouldn't be quite a go. But the hybrids, they had a very nice profile, and especially the H2 and the H1 was the two that the brewery decided are the best candidate to bring forward. So, conclusion of this, we developed this strain. The H2, H3 were selected for a large-scale beer fermentation, and these are the three reasons why: efficient fermentation, optimal sugar attenuation, and different spectrum of flavor. And we were able last August to, to commercialize these beers, which was called Tales from the Future, and it was mature for 18 months in a barrel, and they did infuse a little bit with lemon juice and rose water to make it a bit more zesty and floral. They are a craft brewery, they do these things. I would have kept it as it was, but hey ho. And these are just snapshot pictures from the event. Unfortunately, this beer now is sold out, as literally a couple of weeks ago. And we are now putting out another brew with the same strains for next year. I've got another slide. Yes, it's just a recap. So industrial biotechnology does use microorganisms and enzymes to create bio-based product. So the two main blocks to get to your industrial application is your strain development and your process optimization. In the strain development, so far, the majority of technique are mutagenesis, some strain engineering, where you put things in and out. You got some selection. You put the yeast through, or whatever organism, through a harsh sort of selective procedure, or you do some directed evolution. But I hope that with this talk, I convinced you that we can add harnessing the biodiversity in itself in the strain development procedure. For process optimization, we have done some work on fermentation platform, and Federico has looked at different feeding profile, chemostat versus fed batch versus batch, and how these can actually influence the aroma. And we got something interesting in that too. So, all I showed you about today is pretty much the part of my lab which study evolution in its hybrid for biotechnological exploitation. But I have a big project on functional analysis of non-coding RNA, for which I will give a talk at the conference which I'm going in Puerto Varas. My career really started more on functional analysis and plasticity and genotyping of yeast strains, and on the study of the impact of genomic rearrangements on speciation. And that's that. And my lab has been funded by a bunch of bodies, primarily UKRI, BBSRC, but also NERC and Wellcome Trust for the entire functional analysis, and European Commission for the aroma and biodiversity. And with that, I finished. I just wanted to acknowledge my lab. So this is, when he was taken at that event. There are some of the members now left. Federico went to work for a company. Is it here? I'll just already put that. Where is Federico? Federico went to work for a company in Scotland. He's a senior scientist, fermentation scientist there. Marcin went to work for a genomic company, and is now also head of the genomic facility in this company around Manchester. Javier is postdoc, is PhD of mine, now works with Rike, and some of you might know, and so that's about that. But I mean, this work has been done by them, so I need to thank them. Thank you.
Thank you very much, Daniela, for that great seminar. Going from the Alps.
Change these next time. This is just, I mean, honest.
We will
Yeah.
do something about
Okay.
it. Questions, preguntas. Can be in Spanish, pueden ser en español, pueden ser en inglés.
Yes, just fire.
Sí.
When you make a tetraploid organism, so
Yeah.
when you have twice the DNA of one organism, tetraploid, then you can restore the fertility. But in humans, at least, as I know, having more DNA than you need,
Lethal. In human, lethal.
yes,
Yes.
it may, in the most cases, having more chromosomes may kill the organism.
Yes.
Or produce diseases. How that doesn't affect the hybrid?
Yeah. So yeast in general is really tolerates exceptionally well aneuploidy or polyploidy, or you can have some genome instability. You can grow. I mean, I've not done these, but I'm sure if I take some of my tetraploid and I propagate, I'm sure within 100 generations I will see some chromosome loss, or maybe some rearrangement. We didn't do that because that's they were taken as without any sort of proper selection or propagation. But yeast is very, very resilient to this change of chromosome number or chromosome set. And it is probably more similar to plants. You know, if you think of plants evolution, the plants, several plants evolve just by polyploidization or gross rearrangements, karyotypic rearrangements, and that is a way for the plants to evolve. And actually, it's the only way that we know so far in which you can have speciation and reproductive isolation immediately, because as soon as the plants multiply its genome, it becomes reproductively isolated from the parents, because the gametes, in this case, from the 4N and the 2N, the gametes becomes 2N and N, and with 3N, when you put them together, you're not going to have a correct meiosis. So it is something which is quite more similar to plants, I'd say, in this case, than higher eukaryotes like humans.
Thanks. I have another question.
Yes, right.
The hybrid cell is quite bigger than the cell parent.
So
In one,
hybrid, yeah.
one image, the hybrid were a little bigger than the other cells, the original cells.
The hybrids are bigger.
Bigger, right, yes. Right, again, we're not tested properly scientifically, but I would expect that any organism which has got a double of the chromosome, they would be bigger. Because that happens in other, again, in grapes, we got big grapes because the grapes becomes tetraploid, hexaploid, so the more number you have, the bigger you got your fruit. So I guess, but I've not myself measured the size of the cell, so I cannot really comment confidently on that.
But in general, it's...
In general, you would expect that they're bigger, yes.
One question there. Thank you.
Your group find Saccharomyces ua at high oak trees?
Correct.
Do you believe that's it's not that much common found because do you need like old trees with not that much human intervention on the environment?
In reality, I have not a clue. The first weird thing is that these oaks are 1,000 meter altitude. It's almost unheard. I mean, I don't know how they got there, who put them there, no idea, but there's a little patch of oak. And I sent Johnny there, and that's where he was also able to isolate Uri. So, I really don't know. I don't know. I could not comment much further, but I know Johnny found in some higher oak tree on the Pennines, Uri, and I know that, for example, Brian Gibson found it on I think is Fraxinus, is another type of tree in Bavaria. So tree might matter relatively.
Thanks.
More questions from the audience? So I've got one question.
Yes,
so
right.
You showed us Cerevisiae Kuratsevi hybrids. Why do you choose Kuratsevi as a partner? And no,
Which could it
so for your QTL mapping, you used Kuratsevi. So why Kuratsevi? Why? I mean.
Well, I mean,
other species,
well, we just wanted to do all the hybrids. So we have data for all of them. I just showed Kuratsevi here. But Kuratsevi is the one who my group is dedicating more. We got also Eubayanus and we got also Uvarum that were done by Ed. Also Ed used the selective selection method rather than multi-pool for the QTL, so it was slightly different technique. So. But Kuratsevi is interesting because hybrids are important in wine, and you know, I mean, at the moment with A and B, that company, we have a consultancy, and we created some hybrids between Kuratsevi, the Italian population of Kuratsevi that we found with that project, and we crossed with some commercial wine strain that they gave us, and we found now some hybrids which are really interesting because they produce a lot less sulfite, and therefore they're now being piloted actually for wine. So next year I might even have a wine, not just a beer, a beer and a wine. But yes, so that's the reason why your question is answered if you wanted to know something more about Kuratsevi. But the strain that we chose is the one that were available. But also the other things to say is that these are the things that we put through QTL, but we got a lot of other crossing with different Kuratsevi. I mean, Ed must have done 200 crosses with different things, so we got so many tetraploids that, but then of course when you do a QTL and Illumina, you need to just say, pick something,
¿Alguna otra pregunta? Sí, Eduardo.
Hi. First of all, fantastic talk.
Oh, thank you.
Very interesting.
In spite of that thing.
Yeah, a bunch of different topics, very interesting. I have a question about the H1 and the H2 hybrids. Did you test the genetic stability of the hybrids in time?
Yeah.
Because we have seen some instability even in interspecies hybrids. And I think, related to the previous question, that a lot of instability could be in these hybrids that are about two different species. They are stable over time through different fermentations.
We have not tried, but I think we really should and ought to, because I keep getting this question and I always say we don't try. We ought to try. At the moment, the company asks us a massive pitch of cell, like 10 to the 9. So we produce this pitch, we give it to them, and then off they go. But I think we should really be doing a proper experiment and test again the genome at the end. But we've not done that yet.
Yeah. And the other thing, more a commentary than a question, but I was very surprised to see that when you went to find these different species and the metagenomics, you find Cerevisiae in every sample, basically, in all the trees, in all the altitudes, and I don't know, it's surprising, but I don't know, it's the only species of all that was in all the samples.
I think also Yuri was also a little bit everywhere. The one which really we had much, and I was surprised, is Uvarum. I mean, Ubayanus, fair enough, you know, we were looking for it. We had this 2 OTU at 600, but Uvarum I would have expected to find more, and we didn't see many at all. I mean, almost as little as Ubayanus. While Kudryavtsevi, we couldn't get rid of him. Kudryavtsevi everywhere.
Well, Uvarum and Ubayanus belong to Patagonia.
Yes, we'll see that. We'll see that. We'll see that. No, I mean, I have to say, AB InBev was really interested in finding Ubayanus in Europe, and there is a lot of the sampling that we've done after that, which were pretty much supported financially by AB InBev. And we had a lot of diversity we collected throughout Europe, but primarily, again, primarily we had Uvarum in a lot of different places, but we, of course, know Ubayanus. But AB InBev was very, because personally I think it would be interesting to see what some of these Uvarum are and do, and to sequence some of these, because they were isolated from pretty inaccessible places in Europe. So I think it would be interesting. AB InBev was very much into this Uvarum. I think they were, you know, they just wanted the scoop and find something, and that was that. Yeah, I think the Uvarum is very interesting as a strain, really.
Thank you very much.
Basni.
Thank you for the talk. One of the strains, diploid strains that you generated for beer production, produced at low levels of the spicy flavor. Did you check if that strain lost the genes involved in the production of that?
No, we did for some. So we looked at the STAR1 gene, which is the hyperattenuation one, and basically the uric does not have the STAR1 gene, and the other strain had it in heterozygosis. So when we did the spore-to-spore mating, we were able to cure in the hybrid for that particular gene. And we did also look at the POF, but I think that they were there. They were not gone.
But what hybrid wasn't possible? Hybrid 3 at low levels for vinyl wire code.
One was not vinyl wire code, and the other two they did.
H3, I think the
H3, yeah.
Produced for vinyl wire.
Yeah, it doesn't produce for vinyl wire, but the H3 was the one which was still hyperattenuating too much the beer, and that's why it was not chosen.
So H1 and H2, what percentage of attenuation do they have?
Of what, about 82%, between 80 and 85%, which they liked.
Maybe that strain didn't grow really well, so the spicy flavor.
Yeah, but having said that, after I showed you all these and we published the paper, Brian Gibson was in touch with me because they've done a very similar work, and that is his, it's his expertise. This is again my hobby. This thing is his expertise, and he managed to get quite a decent beer out of just urine, the one got from Fraxinus. So I don't know how, because we couldn't ferment it really properly, because the fermentation would stop, it would not utilize the sugar. So without hybridization, urine wasn't very much. But he has a paper we published a similar time on the beer fermentation with just urine.
Also, Judy is good for bread.
Yeah, I know he did that too, that in the hybrid again.
Yeah.
I think it was also in the hybrid, yeah.
One final question, Javier.
Hola, Daniela. Voy a hacer la pregunta en español porque quiero saber muy bien la respuesta. Si habían probado con el híbrido solo una receta, ¿y cuál receta?
So, did you try just one recipe of beer, or did you try different beer styles?
No, we have tried. So we've done some spontaneous fermentation, but I don't have the data here. And we have, because in that spontaneous fermentation, we meant to do some metagenomic of the population, how they evolved throughout the spontaneous fermentation, but it was spiked with the hybrid. And then they had the barrels fermentation, which is this one. And I think they had also some vessel fermentation, so on a vessel, which I believe again is different. And they explained to me that the barrel was the one which they were more keen to put into the production. So I think
they put some lemon, I saw.
So yes, that's because
beer.
I mean I can tell you they made me taste the beer. And before, and it was really good. And then they put this lemon and some rose petal, and it was too rosy, so I told them. And then, I don't know what they did, but the final product was a little bit less rose petal, but they had, in my opinion, was a little bit too sour, given to the lemon. And personally, I like the beer as it came out first time, just with Yuri straight out, But this craft brewery, you cannot command them, they do what they want. So, I think they had the strain, but then they like to play around with substrate. And the substrate, I still think, has the majority of importance in terms of how the taste comes out. So the yeast is not as important, in my opinion, after seeing how they operate in this brewery, than the substrate they made, they grow this on.
Can I ask another question?
Yeah.
Did you measure the flocculation of the yeast?
No, and I don't believe it flocculated because I would be told. I would have been told, I think.
You would
see it. You would see that they
it's in a barrel. I'm not so sure how. Well,
the barrel is different, but before the barrel, the yeast flocculate.
Yeah, I didn't see any flocculation.
Okay, thanks.
In my family, so, yeah.
Thank you very much, Daniela. Gracias. Aplausos, por favor.
¿Aplauso?
Aplausos.